Hello, everyone. Welcome to the RNA Collaborative Seminar Series. Today is the turn of Zhejiang University's Center for RNA Medicine. So I'm the Executive Director for the center. So I will introduce our first speaker, Dr. Weirui Ma. So Weirui got his PhD from the Institute of Genetics and Development, Biology, Chinese Academic of Science. And then he joined Chris Meyer's lab at the Memorial Sloan Cancer Center for postdoc training. And then at the end of 2020, he joined the Life Science Institute at the Zhejiang University as principal investigator. The primary research interest in his lab is the dynamic mechanism and the functions of cellular RNA localization. So without further ado, let's welcome Dr. Ma's talk on the ribosomal RNAs and localization. Go ahead, Weirui. You can start sharing your slides.
Okay. Thank you very much for Chen's introduction. I'm very happy to have this opportunity to introduce our recent work about the function of ribosome in the organization of the nucleolus architecture. The nucleolus is the largest membranous organelle in eukaryotic cells. You can see here is the HeLa cell under the DSC microscope. Here's the nucleus. Here's the nucleolus. The scale bar here is 5 micrometers. You can see the size of the nucleus can reach 5 microns or even larger. So you can see here that in almost every cell, there is at least 1 or 2 or 3 or maybe even more nucleolus in the nucleus. Basically, the nucleolus exists in almost all eukaryotic cells. The nucleus also plays very important multiple important functions. For example, ribosome RNA transcription happens in the nucleus, and also ribosome biogenesis starts in the nucleus. Recently, it also shows that protein the nucleolus also plays functions in the protein quality control and 3D genome coding. The nucleolus is also a very interesting membranous organelle because it has layered some compartments. During evolution, there are 2 types of nucleolus. One is called bipartite, the other is called tripartite nucleolus. The bipartite nucleolus exists in turtles and in relatively lower organisms such as the fruit fly and C. elegans. It is called bipartite because it has only 2 layered subcompartments. One is called GC. Here you can see under the electron microscope. Another is called FC, fibrillary compartment. In mammalians and birds and also lizard, the nucleus has 3 layered subcompartments, so it is called tripartite. The outside is GC, granular component. You can see on the electron microscope, here's a nucleus of the lizard. This region has those granules, so that's called granular component. This is in the outside. In the center is the FC, it's called fibrillar center. Surround the FC is those electron dense region called DFC, dense fibrillar center. The dense fibrillar has this ring-like morphology on the electron microscope, suggesting that DFC actually has a hollow shell architecture which surrounds the FC. Under super resolution microscope, the typical mammalian cells, the nucleus in nucleus in mammalian cells have a nest-like 3 internal some compartments. Here, NPM1 is a marker for GC, FP is a marker for DFC, POLR1D, which is a subunit of the RNA polymorph 1, is a marker for FC. You can see POL1D forms a small puncture which is surrounded by the FBL ring, which means DFC have a hollow shell architecture which surrounds the FC. So FC and DFC forms functional subunits, which is immersed in the same GC region. Currently, the molecular mechanism of how the nuclear subcompartments formed is not fully understood, and it's also largely unknown that what factors mediates the transition from a bipartite to tripartite nucleolus. Recent years, there are lots of progress has been made in the formation of a nucleolus. For example, liquid-liquid phase separation has been proposed as a mechanism for the formation of nucleolus. And recently, a very beautiful work from Cliff Brown's lab shows that the nucleus can be viewed as a multi-phase liquid condensate because the different some compartments showed dynamic exchange with the surrounding, which means they have those liquid-like property. So in the nucleus there are multiple some compartments, but they are not fused into a uniform phase. So here is the question: if those different subcompartments are liquid-like droplets or they have liquid properties, why don't they just fuse into a uniform phase? In this work, Cliff Brown shows that this in vitro and in vivo constitution, this shows that actually it's the difference in the droplet surface tension prevents the fusion of the different compartment. For example, FIB1, which is a homologue of FDL in human, and NPM1 is the marker for GC. FIB1 forms those solid droplet inside the NPM1 droplet. The surface tension of FIB1 and the surface tension of NPM1 is very different. Therefore, they cannot emerge. They are immiscible. This work is very beautiful because it demonstrates a principle of the formation of how this core shell structure. Also, the method used in this study, the in vitro reconstitution, is a very powerful approach to study the organization of the nucleus. However, you can see here that that's the nucleus under super resolution microscope in mammalian cells. The FBO, which is marked for DFC, forms this ring-like structure. In this reconstitution, FIB1 forms solid droplet. So this still largely unknown. How is the hollow shell architecture of DFC formed? What factors are critical for the formation of the hollow shell architecture of DFC? We think this architecture is really might be very critical for the formation of tripartite nucleus because this compartment separates the FC and the GC region. If there's no DFC, of course, there will be only two compartments. When I was a postdoc in Christy Mayer's lab, we found that RNA interactions is able to regulate the morphology of RNA granules. For example, here is a fast-twist protein. Fast-twist protein forms a sphere-like droplet without RNA. However, when specific RNAs are added into the phase separation system, those three RNAs, but not fast 3' RNA, are able to induce mesh-like condensate. This work demonstrated that we further demonstrated that RNA interactions are critical for this. So after I established my lab, I was wondering what if RNA also plays important roles for the formation of the nucleolus. three-layer compartment. In order to answer this question, we first did imaging to visualize how does RNA look like inside the nucleolus. We use PI, propylialdehyde, to stun RNA. PI is a intercalating dye which combines to DNA and also double-stranded RNA. You can see here that we use DAPI to stun DNA, PI to stun both DNA and RNA. That's a typical nucleolus in several different cell lines. The DAPI signal in the nucleolus is low, but the PI signal is high. And after RNA treatment, the PI signal is largely decreased, indicating that in the nucleus, the PI signal is mostly from the RNA. And on the super resolution microscope, we found that very interestingly, those RNAs, the distribution of the RNA in the nucleus is not uniform. They form some kind of like mesh-like assemblies and with those hole-like structures inside the nucleus. Those hole-like structures just remind us the DFC and FC function subunit. Then we did a co-stunning with FBL, which is a marker for DFC. You can see here that those hole-like structure is where the DFC localized. Inside the DFC FBL ring, which is FC, RNA is largely excluded from the FC. The RNA localized mostly in the GC region and closely surrounds associated with the DFC. This data is very interesting and also suggested that RNA maybe is very critical for the formation of those structures. In order to answer this question, we used RNA-seq experiment. We did RNase experiment. RNase-L is a RNase, but in cells it's not active. Only when double-stranded RNA are present in cells, which, for example, virus infection or bacterial infection, RNase-L will be activated. We use poly-IC transfection, which is analog of double-stranded RNA to active RNase-L. We first fused the RNase-L to BAP and to a nucleolus localization signal to drive the localization of RNase-L in the nucleolus. Here, for example, in this cell 2. Then we transfected poly-IC to cells to activate RNase-L. You can see here that cell 1 is a cell without RNase expression. After poly-IC transfection, the FBO still forms those run-like structure, indicates the DFC have a hollow shell architecture. However, in cell 2, which has nuclear localized RNase-L after poly-IC transfection, the run-like structure of FBO is largely decreased, and the FBO forms this solid droplet. This status indicates that RNA is essential for the formation of run-like morphology of the DFC. So next, we would like to know, because in the nucleolus, there are several different types of RNA, including ribosome RNA and the SNO RNA and the other RNAs. We would like to know which RNA is critical for the formation of run-like morphology of DFC. Because ribosome RNA is the most abundant RNA in cells, it takes about more than 80% of total RNA, and the ribosome RNA transcription happens in the nucleus. Then we think, oh, it should be the ribosome RNA are critical. It's critical. In order to answer this question, we first did RNA fish fluorescence in situ hybridization to visualize the ribosome RNA in the nucleolus. In human cells, there are 4 mature ribosome RNAs, 5S, 18S, 5.S, and 28S. 5S is transcribed by RNA polymerase 3 in the nucleus, not in the nucleolus. But 18S, 5.S, and 20S is transcribed by RNA polymerase 1 in the nucleus. It is transcribed as a same precursor pre-ribosome, then it is cleaved into the mature form of those 3 RNA. Then we designed 4 probes. One is targeted to the 24S region, to the 5 prime ETS, which specifically detects the ribosome RNA. Then those 4 localized to the mature region of the ribosome RNA. Then we performed RNA fish under super resolution microscopy. We first did 5 prime ETS because it is published, it is reported that ribosome RNA transcription happens in the FC and also the interface of FC and the DFC. Then what we observed is that we often saw those bright dots localized just inside the DFC run, which is the FC. This is we think that's the transcription side. This is consistent with the previous data. So which means our probe, fish experiment on our super resolution microscope works. Then we did 5S, 5.S, 18S, and 28S. 5S ribosome it is transcribed in the nucleus, and it is not enriched in the nucleolus. 5.S, 18S, and 28S. In the nucleus, it is highly enriched to the nucleus. And under super resolution microscope, we saw that 5.S actually is enriched to the FBL root, which means it is co-localized with FBL root, which means it is enriched to the DFC some compartment. However, 28S and 18S is not enriched in the FBL run. I would like to point out that for 28S RNA, it is very interesting. It looks very similar to the PI signal because in the FC region, which is here, the 28S RNA is largely excluded and is enriched in the GC region. But in the DFC region here, there's still very high signal of 20S RNA, which means 20S RNA localized primarily in the GC and also DFC region. For me, this data is very interesting because those 3 ribosome RNA are cleaved from a single precursor mRNA. Why do they have different localizations? Currently, we don't have the answer. Because in cells, in human cells, there are hundreds of copies of ribosome RNA, and there are millions ribosome RNA genes exist in the genome as tandem repeats, and there are millions and millions of copies of ribosome RNA. So it is impossible to knock out or to knock down the expression of ribosome RNA. But we would like to know, are those ribosome RNA really responsible for the formation of DFC? Therefore, we employed the in vitro reconstitution experiment. We purified BIP-tagged NPM1 and the GIP-tagged FBO, which can undergo phase separation in vitro, just as published. Then we also cloned the full-length ribosome RNA genes and did in vitro transcription to synthesize those full-length ribosome RNA. You can see that's the total RNA. That's 18S, that's 20S. The size of our in vitro transcribed ribosome RNA is similar to the 20S and the 18S RNA in cells, which inform their full length. Then we did the in vitro reconstitution separation experiment without or with ribosome RNA. Without ribosome RNA, NPM1 and FBL forms this spherical droplet, very beautiful. The fluorescence signal the distribution is relatively uniform. However, when we added 3 types of ribosome together into the system, when the ribosome reaches the very critical concentration here, 1 micromolar, which is about 2.3 microgram per microliter, then we observe the formation of those ring-like structures is similar to the DFC structure in cells, and inside the ring-like structure of FBL ring, the NPM1 is largely excluded. This is also similar to the FB and MPL1 signal in cells. So we think that we are very happy about this. We think that's the DFC-like structures, which means ribosome RNA is able to induce the formation of DFC-like structures in vitro. I would like to point it out that in our in vitro reconstitution experiments, not every droplet forms DFC-like structure. There are only about 20% of droplets, especially of those with large size, uh, uh, has a DFC-like structure. Which means those droplets with small size, there's no DFC-like structures. We currently don't know why. Probably because in those large droplets, there are more ribosome inside. Because in our reconstitution, we added 3 types of ribosome, then we would like to know which one is the most critical. Then we did the combinations. What we found is that if we remove, only when we remove 28S RNA, those DFC-like structures were not formed, and if we just only add 20S without 5.S and 18S, just 20S alone with FB and MPL1, this the DFC-like structure can be induced. So we concluded that the 20S RNA is very critical for the induction of DFC-like structures in vitro. So next we would like to know if only FBL with 20S or MPL1 with 20S is sufficient to induce the DFC-like structure. So we just mixed FBL alone. This is FBL alone or FBL with 28S. This is MPL1 alone. This is MPL1 with 28S. There's no DFC-like structure formation. Only when those 3 components exist can induce the formation of these DFC-like structures. This data suggested that interactions among the GC component, the DFC component, and the 20S RNA is essential for the formation of DFC-like structures. Previously, we showed that RNA interaction is critical for the formation of match-like condenses. So here we would like to know what is RNA interaction of 20S RNA play roles in the formation of DFC-like structures. So first, we did RNA interaction experiment by simply just annealing of 20S RNA, and then we run a native agarose gel. You can see after annealing and in the native agarose gel, 28S RNA forms this very high molecular weight species, which means 20S RNA indeed has the ability to form intermolecular RNA interactions. So we can say 20S RNA is a multivalent RNA. Then we'd like to know if this interaction happens and this interaction is indeed required for the induction of DFC-like structures. Previously, a beautiful lab in Ronville's lab shows that CG repeats RNA is able to phase separation in vitro through RNA interactions, and doxorubicin, which is a nucleic acid intercalator, can disrupt the base pair interactions when the doxorubicin is added into the in vitro phase separation system and blocks the formation of blocks the CG repeats RNA phase separation. So we also added doxorubicin into our in vitro reconstitution experiment. You can see after adding of doxorubicin, we will observe that the DFC-like structure is largely decreased, which this data suggested that multivalent interactions in 20S RNA may be required to induce the formation of DFC-like structures. I mentioned in the introduction part during evolution that 2 types of nucleolus. One is tripartite, the other is bipartite. Then we would like to know is it is possible that 28S ribosome is a candidate to mediate the transition of bipartite to tripartite. In order to answer this question, again, we cloned the 28S RNA from different genes. We cloned from mouse and chicken, which the nucleoli are tripartite, and also cloned 28S from fly, fruit fly, and C. elegans, which is the nucleus is bipartite. This is the agarose gel. Also, we did in vitro transcription to synthesize those ribosome RNA. Then we did the interaction experiment again. You can that's the native agarose gel. It's very interesting that a human mouse and as the quantification, a human and chicken, the ribosome RNA shows the highest RNA interaction are the fly and C. elegans, which is the bipartite nucleus, which is the lowest RNA interaction, which means 20S ribosome RNA acquired multivalency during evolution. And then we did the in vitro reconstitution experiment again. Very interesting, you can see here that 28 ribosome RNA from human mouse and chicken can induce the formation of DFC-like structures, but 28 siRNA from fly and C. elegans cannot. Then we did some very simple analysis about the sequence of those 28 RNA because we would like to know what the features drive the contribute to the multivalence of 28 RNA. What we found is that actually ribosome is actually very conserved, especially for 5.S and 18S, but 20S compared to 5.S and the 18S, it is not as conserved as the. For example, in mouse, 18S is about 100% identity, but 20S is only about 22% identity, and the length of 20S ribosome undergoes substantially lengthening, and the extra region in human mouse or chicken are usually GC rich. GC rich, you can see here that as the extra region compared to human C. elegans is almost for GC. So we propose that lengthening and high GC content may contribute to the multivalency of 28S ribosome RNA. At last, we would like to know what if 20S RNA is able to really induce three-layered nuclear GC-DFC-like structures. So in order to answer this question, we provide one more protein, POL-R1D, which is the component of FC in cells. Then we did the in vitro reconstitution again. Again, you can see here that 28 siRNA from those organisms can induce these three-layered GC, FC structures, but 28 RNA from fly and C. elegans cannot. Okay, that's the take-home summary of today's talk. That's the take-home message, which shows that with super resolution microscope, the ribosome RNAs have specific localization in the nucleus. And we also found that 20S siRNA is able to induce DFC-like structures in vitro. And we propose that 20S RNA is a candidate that mediates the transition of the bipartite to tripartite nucleolus. That's in the end, I would like to thank our lab member. This work is largely is almost done by Yiwi in my lab, and I would also like to thank my collaborator Chen Qixiang and his student Zhejiang purify the proteins. Thank you much for your attention. I'm happy to take questions.
Okay, thanks, Weirui. So to our virtual audience, please add your question for our speaker in the Q&A. So because we almost reached the time, so I will just start with a quick question, Weirui. So you do your annealing experiment. So I'm a little confused. So basically you use the in vitro synthesized RNA and anneal with the total RNA or anneal with itself?
Itself.
Okay. So you're showing that the RNA from the tripartite organism is more likely to form more complex structure. That's what you meant, right?
Oh, no secondary. It's from intermolecular RNA interaction.
Yeah. Okay.
Yeah.
Intermolecular RNA.
Yes.
And also I saw like you did a different titration of to show in the droplet to show the structural formation. So I saw you reach the a critical concentration. Did you try
Yes.
what if it's go higher? And here you show one. What if it's go higher?
Oh, you mean the concentration in cells?
Yeah.
Oh, actually, the concentration used in our in vitro reconstitution is lower than in cells because the solubility of 20S becomes uh, uh, not very good when the concentration is very high. So we cannot increase the concentration of ribosome RNA in our in vitro reconstitution. In cells, it's about 5 microgram per microliter based on some publications.
Okay. Okay, here's a question from Cao Dong. So he said, like, I wonder whether do we know whether the multi-layer structure are important for nucleolar function? Is the protein that from the hollow structure essential?
Oh yeah, yes, the nucleolus multi-layered architecture is essential for I cannot say essential, but it's important for its function because a very beautiful work from Lin Chen's lab shows that uh, a non-coding RNA is able to regulate the size of the DFC run, then also regulates the uh, efficiency of ribosome RNA transcription. So the structure is, of course, is important. Yeah.
Okay. Okay, then thanks, Weirui. So I think you can stop sharing and I will
Okay.
introduce our, thank you. So I will
Okay. Thank you very much.
introduce our second speaker, Chen. So Dee got his PhD also from the Institute of Genetics and Developmental Biology, Chinese Academic Science. And then he joined Professor Amanda Clark's lab for postdoc training at the UCLA. So at the end of 2019, Dee joined the joint institute between Zhejiang University and the University of Edinburgh as assistant professor. So his lab focused on post-transcriptional regulation of the differentiation of human embryonic stem cell. Now he will tell us, in his talk, he will tell us about the M6A's regulation in this differentiated human embryonic stem cell. So welcome, Dee, so go ahead.
Okay, thank you, Xin. I really appreciate for your kind invitation and also the kind introduction. And I'd also like to thank the organizers for giving me this great opportunity to share my recent work. So I'm turning my camera off. Give me 1 second. Okay. It looks like I can't turn it off. Okay, that's good. Okay, cool. So as Xin mentioned that I'm focusing on the post-transcriptional regulation of the differentiation of human embryonic stem cells. And today I will talk about our recent progress in M6A encoded regulation in human genetic development. So germ cells are the only cell type in our body that pass to our next generation. They're the carriers of genetic and epigenetic information. And in humans, they are specified as early as about 2 weeks during the development. So actually, 2 weeks is kind of awkward time during development because we don't have access to the human development at around 2 weeks. So basically, we know very little about how the germ cells or how the germline is established during development. So after the precursors of the germ cells are specified, and these precursors are called primordial germ cells, or PGCs for short. And these PGCs, after specified, they will migrate and they will meet their somatic partners to form the gonad, and eventually they will form the mature gametes, sperm and egg, and the fertilization of the egg by the sperm will start a new life again to make the life cycle. And we are particularly interested in how the germline is established and how this process is regulated at the post-transcriptional level. And during my postdoc training at Amanda Clark's lab at UCLA, so we established in vitro system to study how the cells are specified because it's really hard to work on the human embryo at this time point when the germ cells are specified. So we basically take advantage of the cultured human embryonic stem cells, which are derived from developing human embryo. And these human embryonic stem cells, they are pluripotent, which means that they have the potential to differentiate into most cell types in our body, including germ cells. So when I was a postdoc, so we established the 3D culture system to induce the germ cell-like cells in vitro and to study how the germ cell phase is specified. So basically, the system we use is like this cartoon shows. So we differentiate the embryonic stem cells, which is HESCs for short, to some kind of intermediate cells and then to the 3D culture in this low attach 96-well place. And that's day 4 of induction. So we were able to induce about 10% of the cells as the primordial germ cell-like cells, HPGCLC for short. And using this system, we have discovered a lot of the transcription factors that play critical roles for the induction of the germ cell phase. But actually, we know very little about how the post-transcriptional regulation is involved in this process. And when I established my lab in Zhejiang University, so we are particularly interested in the M6A modification mediated post-transcriptional regulation. And for the M6A modification, they have writers which add the method group into the mRNA, and also they have the erasers to remove the M6A modifications. And also these M6A modified RNAs are recognized and bound by the M6A readers, and the different readers may trigger different downstream effects. So for example, some readers are important for the translation of the mRNA, and some readers are important for the stability, either for decay or stabilization of the mRNAs, and some readers are involved in the splicing of pre-mRNAs, and some readers are involved in the nuclear exports of the mRNAs. So they're involved in a lot of aspects of RNA metabolism. So we're wondering if this M6A pathway is involved in the regulation of the specification of human germ cell phase. So first we check the expression of all these writers, erasers, and also readers for the M6A in embryonic stem cells, and also different kind of early embryonic cells, including the germ cells. And we found that all these writers, erasers, and readers are highly expressed in almost all cell types, indicating that the M6A pathway may be important for all this process. So actually, at the very beginning, we expected to see some cell type specific expression of the either the reader, eraser, or writers, but we didn't see the cell type specific expression pattern. So we try to inactivate the M6A pathway and to determine or to detect whether M6A pathway is involved in the regulation of the human germ cell development. So we started with the writer method 3, so which is one of the most important writer protein for M6A modification. So we made the knockouts ESC lines using the CRISPR-Cas9 knockouts. So in order to exclude the possibility of the redundancy function or the other incomplete loss of function, so we knock out the whole genomic region of the method 3 by using a pair of gRNAs to knock out the whole genomic region. And we made these mutant embryonic stem cell lines, and we found that morphologically, the mutant cells are similar to the control cells, and we use Western blot and also qPCR to verify that indeed the method 3 gene is deleted. We also verified the results by immunofluorescence, and we can see that the pluripotency gene OX4 is expressed in the mutant cells, but method 3 gene is completely knocked out. So next, we wonder whether method 3 knockouts will impair the development of the germ cells. So we use the 3D culture system to induce the PGCLCs. And at the day 4 of the germ cell induction, we found that in the control cells, that about 7% of the cells are the germ cells using integral alpha 6 and epicam as surface marker to label the germ cells. However, when method 3 is knockout, so we discovered about 2-fold increase of the PGCLCs, suggesting that method 3 may be involved in the suppression of the germ cell phase. And also, we overexpress the erasers because for writers, they add the M6A, and for erasers, they remove the M6As. So in order to prove that the M6A pathway is involved in this process, we remove the M6A by overexpression of the erasers. There are 2 eraser proteins for M6A. One is the ALKPH5. So when we overexpress the ALKPH5, we discovered the increased induce of the human germ cells, just like the writer knockouts. We also overexpress the FTO, another M6 eraser, and we discovered the same trend. So we can also see the increased induction of the PGC-like cells from about 10% to about 15%. So which means that M6A pathway is probably involved in inhibition of the PGCLC induction from the embryonic stem cells. So next we wonder which M6A reader or readers are involved in this process. So as I mentioned, there are different readers for the M6A mediated regulation, and actually the M6A pattern is different for different cell types, and the cell type specific pattern is mostly determined by the balance of the writer and the eraser of the M6A machinery, but how the M6A encoded information is recognized and also interpreted in the cells to regulate cell phase is largely determined by the reader proteins, because these reader proteins, they bind to the M6A modified RNAs, and also they interact with different protein partners to mediate different downstream effects to the mRNAs to regulate the phase of the mRNA and eventually regulate the phase of the cell types. So we are wondering which reader is involved in the regulation of the human germline. So first we check the expression of these readers, and we didn't see the cell type specific expression of the reader. So we performed a small scale CRISPR-I screen to detect which reader is probably involved in this process. So so in order to make it short, so I will just mention that the knockout cells and the knockdown cells by CRISPR-I is labeled in red, and the control cells is IOP negative. So we mix the cells together and induce them for the PGC-like cells, and using flow cytometry, so we were able to tell which cells are knockdown cells and which cells are the control cells. And we normalize the percentage of the PGCLCs in the knockdown cells to the control cells. So we can see that if we normalize the control cells to 1, we can see that 2 of the readers, especially for this IgF2BP1, displays the same phenotype as the knockouts of the method 3, so which is the increased percentage of the PGC-like cells. So next, we focus on this IgF2BP1, and in order to confirm the phenotype, so first we designed a pair of gRNA to delete the whole genomic region of the BP1. BP1 is a little bit larger compared with method 3, so we use the donor replacement strategy. In this way, we put the drug resistance gene to replace the whole genomic region of the method 3. So for example, if we put the puromycin here, so it's easier for us to screen out for the knockout cells by drug selection. So after we obtained the BP1 knockout cells, just like the method 3 knockout cells, morphologically they're very similar to the control cells, and then they can be maintained for passages and passages. Using Western blot and qPCR and also immunofluorescence, we confirmed that indeed the BP1 protein and the BP1 gene is completely depleted. And also we induced germ cells from the BP1 knockout embryonic stem cells, and we discovered the same phenotype increase the percentage of the PGC-like cells. So for example, in control, it's about 10%, and in the BP1 knockout, it's about 20%, about twofold of the increase for the PGC induction. And also, in order to check the cell phase of these PGC-like cells, we isolated these cells by FACS using the cell surface marker of integral 6 and also EpiChem. When we performed RNA sequencing of these control PGC-like cells and also these mutant BP1 knockout PGC-like cells, we found that they are clustered with each other. So and there are very few differentially expressed genes. So which means that although the percentage of the germ cells increased, the germ cell phase is not altered. So BP1 and the M6A mediated regulation is probably more involved in controlling the induction of the PGC-like cells, but not the development of the germ cells. In order to perform that, this regulation is M6A dependent. So we try to rescue the phenotype of BP1 knockouts by overexpressing either the whole net, the full net of the BP1 gene, or the truncated form that's KH3 and KH4 domain are truncated, and they fail to recognize the M6A modified RNAs. So in the control, so you can see about 5% of the PGC-like cells, and when BP1 is knockouts, there is an increase of the percentage of the PGC-like cells. When we overexpress the full-length BP1, so we can rescue the phenotype to about the control level. However, when we overexpress the truncated form, so which doesn't recognize the M6A modified RNAs, they fail to recognize to rescue the phenotype, suggesting that BP1 regulates the induction of the PGC-like cells through the M6A recognition. So to summary this part of work, so first we discovered that M6A pathway is involved in inhibition of the PGC LC induction from human embryonic stem cells. And also we discovered that the M6A reader IgL to BP1 is involved in regulating this process. So it suppresses the induction of the PGC-like cells from the embryonic stem cells. So next we wonder how BP1 regulates this process by identification of the RNA targets bound by the BP1. So we reasoned that because BP1 is a M6A recognizing protein, is a reader of the M6A, the key downstream targets of the mRNAs for the BP1 should be modified by M6A. So we performed M6A-seq in human embryonic stem cells. And also the key downstream targets of the BP1 should be bound by BP1 itself. And also in previous work, it has been discovered that BP1 is involved in stabilizing the mRNAs. So when BP1 is not caused, the key downstream gene should be downregulated because BP1 stabilizes. Without BP1, it will be degraded. So we overlap these three categories of the RNAs, and we found that there are 7 genes match this criteria. And for these 7 genes, we performed a small-scale CRISPR screening, and we found that only for the first gene, OTS2, it displays the same phenotype as BP1 and MED3 knockout, which is increased the percentage of the germ cells. And for this OTX2, it has been discovered in mice that it regulates the germline entry of the PGC in mouse. And when OTS2 is mutated, the germ cells is increased, but the molecular mechanisms about how OTS2 is involved and also how OTS2 is regulated is largely unknown. So we use this same system and try to understand how BP1 acts through OTS2 to regulate the germ cell development. So first, in order to prove that indeed OTS2 functions downstream of the BP1, we try to rescue the phenotype of the BP1 by overexpressing OTS2. So as expected, there's about 10% of the PGC-like cells in the control, and when BP1 is knockout, is increased to about 30%. And when we overexpress the OTS2 in BP1 knockouts, we can rescue the phenotype. And also when we check the expression level of the RNA, so we can see that in a BP1 knockouts, the OTS2 RNA is decreased in both embryonic stem cells and also these intermediate cells here before we made the germ cells. And when we overexpress the OTS2, so we can see the increased level of the OTS2 and also the rescue or the phenotype. So suggesting that OTS2 functions downstream of the BP1. And also in order to provide the direct evidence that BP1 binds to the M6A modified OTS2, we also check the M6A6 data in embryonic stem cells in a gene region of the OTS2, and we found that compared with the input control, so we can see this M6A modification in a gene region, especially the 3' UTR region of the OTS2, and also when we perform the BP1 eclipse, and then we can see that the enrichment of the BP1 protein in this M6A region, and also the 3' UTR region of the OTS2. And also we confirmed this binding of the BP1 to OTS2 3' UTR by RIP qPCR experiment. And by this RIP experiment, so we designed the PCR primers to these regions based on this eclipse data. And we use the BP1 antibody to perform the RIP qPCR in human embryonic stem cells. And we found that indeed there is an enrichment of the BP1 in this region. So suggesting that indeed BP1 binds to the M6A modified region in the OTS2 mRNA. And also in order to prove that indeed the binding of the BP1 to OTS2 3' UTR is M6A dependent and also is critical for the regulation of the stability of the OTX2 mRNA. We designed the following experiment. So as I mentioned before, that the KH3 and the KH4 domains are critical for binding of the BP1 to the 3' UTR OTS2. So this full-length BP1 binds to the OTS2 RNA, but the truncated form, it doesn't bind to the M6A modified 3' UTR of the OTS2. And we also polluted previously that this one doesn't rescue the phenotype of the BP1 knockouts. So we took advantage of this truncated form of the BP1. So as in theory, so BP1 binds to the M6A modified 3' UTR of the OTS2. If we put this 3' UTR OTS2 downstream of the luciferase gene, so we can use the luciferase assay to determine how BP1 may regulate the OTS2 mRNA. So in this condition, in the wild type BP1, it binds to the M6A modified RNA. In the truncated form, it won't bind to the RNA. But if we use the MS2, which is a RNA element that could be bound by the MCP protein. So if we put the MS2 be upstream of the OTS2 3' UTR, and we put this MCP and the BP1 truncated fusion protein, so we can tease this truncated form of BP1 to 3' UTR of the OTS2 downstream of the luciferase, and in this way, in theory, it will activate the stabilization of this RNA. So indeed, by luciferase assay, we can find that as BP1 stabilize the RNA, so it leads to the increased luciferase activity compared with the control. But if we use the truncated form of the BP1, it won't activate this one, activates the luciferase activity. But if we use the MCP and MS2 to tether this truncated form of the BP1 to the 3' UTR, it's also increased the luciferase activity. So which means that indeed the binding of the BP1 to the M6A modified RNA is critical for stabilizing of the OTS2 RNA. And also when we overexpress the BP1, we found that it leads to the increase of the OTS2 mRNA and the decrease of the percentage of the germ cells. So, which means that BP1 is critical for regulating the mRNA level of the OTS2, and the OTS2 level is critical for the germ cell development. So, to summarize these parts, so we found that the OTS2 mRNA, when it is transcribed, it will be degraded if there's no M6A modification by the M6A writers. And if the writers added the M6A modification, if there's no M6A reader, it will also be degraded. But if there are M6A readers, and in our case, it's the adult BP1, this reader binds the RNA to stabilize the LTS2 mRNA and to stabilize the RNA to promote the translation of the OTS2 protein, and the OTS2 protein in turn will suppress the induction of the PGCLCs from the embryonic stem cells. And our next question will be how OTS2 suppress the induction of the embryonic stem cells. So we performed the co-IP mass specs and try to identify the partner of the OTS2 in the embryonic stem cells, try to identify which could be the potential downstream the partner of the OTS2 in regulating the germ cell development. And we found that TFAP2C is one of the key factors that's physically interacts with OTS2, and we confirmed this physical interaction by co-IP experiments. And we also checked the expression of the OTS2 and found that it's highly expressed in this intermediate stage. And we performed the chip sequencing of the TFAP2C and also cut and tag the OTS2, and found that the binding patterns is very similar for OTS2 and the TFAP2C, and also they physically interact with each other, suggesting that OTS2 may suppress the TFAP2C in regulating the germ cell development. So for my whole career of postdoc, I've worked on this gene TFAP2C and about how TFAP2C regulates the development of human germ cells. And we discovered that TFAP2C is a critical gene for germ cell development. Although germ cells are specified at this stage, TFAP2C is already expressed in the embryonic stem cells, and also it binds to the germ cell genes that are not expressed in the embryonic stem cells. So which means that at early stage, TFAP2C binds to these genes, but not expressed, and in the germ cell stage, it binds to the germ cell gene and it's expressed. But we didn't know at last time why the binding of TFAP2C doesn't lead to the expression of the germ cell genes. So by in this project, we think that probably OTS2 is the one that suppress the function of TFAP2C at these early stages. So in order to prove this hypothesis, so we use the gene SOC17 as a reporter to test whether the binding of TFAP2C could promote the expression of SOC17 and whether the co-expression of TFAP2C and OTS2 will suppress the expression of the SOC17. So we cloned the promoter of the SOC17 to the luciferase reporter, and we found that overexpression of TFAP2C indeed promotes expression of this SOC17 promoter. However, when we co-express the OTS2, it suppresses this activation. And also, in order to prove the genetic relationship between OTS2 and the TFAP2C, we performed the double mutants experiments. So we made the double mutants for TFAP2C and OTS2, and we used the immunofluorescence to prove that indeed in OTS2 mutants, there's no OTS2, and in TFAP2C mutants, there's no TFAP2C, and then in double mutants, there's no these two proteins. And they found that in the control is about 5%, and OTS2 is an increase, and in TFAP2C, there's no germ cells. And if TFAP2C functions downstream of the OTS2, the double mutants will display the same phenotype as TFAP2C. And indeed, we see that there's no PGC-like cells, just like the TFAP2C, indicating that indeed TFAP2C functions downstream of the OTS2 in regulating human germ cells. And lastly, we try to prove by more evidence that indeed the BP1 regulates the M6A modified OTS2 to regulate this process by designing the DCAS13B experiments. So we use the M6A eraser, ALKBH file, and push them to the DCAS13B, and then use the cRNA to push this complex into the OTS2 specifically. So which means that using this tool, so we remove the M6A in OTS2 mRNA specifically, and we try to examine what happened to the germ cells. And we found that indeed, when we remove the M6A modification specifically in OTS2, we found the same phenotype, increased human germ cells induced from the embryonic stem cells. So confirm that indeed the M6A modification is critical for the regulation of the human germ cell development. So this is our working model that LTS2 should be modified by M6A and recognized by the M6A reader IgO to BP1 to stabilize RNA, and in this case, the OTS2 protein can be expressed, and OTS2 protein will suppress the function of TFAP2C to suppress the induction of the human germ cells. And I think this braking system is important. So we know a lot about the positive regulators of the germ cells, that when they are mutated, the germ cells are gone, but they're very limited discoveries about these inhibitors. So I think this is very important because during early development, although germ cells are development, we don't want too many germ cells because if there are too many germ cells at the expense of the embryonic development, this will lead to the embryonic development defects. Eventually, it may lead to the birth defects. So that's all for my sharing. And I would like to thank all the students in the lab, especially Jing, who did most of the work for this project. And also I'd like to thank our collaborators and our fundings. So thank you for your attention, and I'm happy to take any questions.
So thanks, Dee. So just for the audience, please, if you have questions, please type your questions in the Q&A. So Dee, I may start because you are using a system pretty powerful to induce the PGC from ESC. So in this system, so when you do knockout, do you or expression, do you select a single colony to do this kind of experiment?
Yes, we have to, because it's so for the human embryonic stem cells, actually the knockout efficiency is quite low. It's less than 5%. So most of the time we need to purify the single colony, and sometimes we need to purify multiple times to make sure that indeed it's homogeneous for the mutants.
And for the overexpression, so what kind of system you're using? So what promoter you're using for the overexpression?
So for the overexpression, so we use the test-on system. So which means that when we add the drug, we trigger the overexpression, because sometimes a lot of gene expression will lead to the cell phase alteration in the stem cells. So we don't want to overexpress genes constitutively in the stem cells. So mostly we use the test-on system. So which means that if we don't add the drug, it's just like the control or like the mutants, and when we add the drug, it will overexpress gene of interest.
So, but that would integrate into a random location or in a specific location when you
Random, not
okay,
not the
okay.
location. We use an antivirus.
Okay. And also for the M6A loci you identified in the BP1. So do you see whether there's any conservation of that locus? For the M6A modification locus, because normally it has a motif, right? So I wonder if you look through the evolution, whether that is a conserved one.
Yeah, it's GGACU. It's the one of the classic one. And actually there are multiple sites. We still don't know which one is probably the most important one. We have identified 4 M6A sites that are critical in this region.
So there's a question from Madeline Mayday. So her question is, maybe I missed this. Do you see an increase of OTX2 in metal 3 knockout cells, or is this solely depend on the readers?
Okay, so I didn't put the data here. So actually in metal-3 mutants, we see the decrease of the OTX2 mRNA, just like the BP1 knockouts. So we think that if there's no M6A modification, this RNA is not stable.
Okay, I hope that addressed the question. Okay, I think we would like to, time is up, so we have to conclude here. Let's thanks Weirui and Dee again, and also thanks all the audience. Okay, we will see you like in the next seminar series. Okay, bye.
Thank you. Bye.